In this mini lecture, we're going to talk about lymphocytes and their major roles. There's also a whole separate video reviewing T cell activation that's uploaded and available for you to watch. The immune system is a very complex topic, as you're probably figuring out. There is this whole section in the chapter of your book on clonal deletion, which is the selection process that T cells undergo. But we are not going to be talking about that here. If you're interested in how you select for the right kind of T cell, you're welcome to read that part of the chapter. T cells, as I mentioned, are made in the bone marrow and they learn how to be a T cell in the thymus. Once mature, they're going to be circulating in the lymph and in the blood. They congregate in the lymph nodes, spleen, Peyer's patches, and MALT and GALT in mucosal tissues. Those spots are where they're often going to interact with cells that are presenting epitopes to them. The epitopes are going to be presented on MHC. And remember, MHC1 is on all cell types, and MHC2 is on our specific phagocytic cells and B cells. The MHC presenting those epitopes are going to interact with the T cell receptors on the T cells. However, the T cell receptor is also going to be specific for the particular antigen. In this picture on the bottom left, you can see a killer T cell with its T cell receptor interacting with an infected cell, which must have had an endogenous antigen presented with MHC, which in this case would be MHC1. The epitope is shown in yellow here. That is how the cells interact with and talk to each other. Now, T cells are very interesting, and again, we're not going to spend time discussing the details, but there's changes that are happening at the DNA level that will allow the T cell to actually rearrange their DNA to make functional genes for the T cell receptor. They have what you can think of as little cassettes that combine and allow our T cells to have about 10 to the 9th different combinations to generate different T cell receptors. And that's pretty amazing. A diversity of T cell receptors is key to recognizing all the different possible antigens out there. What is different or unique about them is the antigen binding site that allows them to interact with lots of different epitopes. As mentioned already, we have different types of T cells. The cytotoxic, or otherwise known as killer T cells, have a marker on their surface that identifies them, and that cell surface molecule is called CD8. Often, you'll hear about cells being CD8 positive. That just means they're cytotoxic T cells because they have that CD8 molecule on the surface of the cell. The helper T cells are CD4 positive. Remember from that video clip on the HIV life cycle? CD4 is a receptor for HIV as well. You find CD4 on helper T cells. The helper T cells are the coordinators of our immune response. They help both B cells and cytotoxic T cells do their job. Because we have those two specific roles, we actually have subtypes of helper T cells. T helper type 1 cells are the cell type that help the killer T cells. And the T helper type 2 are the ones that help B cells. They are both helper T cells. They just secrete different chemical signals to either stimulate a killer T cell or to stimulate a B cell, depending on what subtype of helper T cell they are. We also have regulatory T cells. We're not going to be spending time talking about them. They have different CD receptors on the surface, including CD25, and their role is to help tamp down the immune responses so that we don't respond inappropriately to antigens. In short, the T cell receptor, or TCR, of a helper T cell is to enable the interaction with MHC2 with the help of the CD4 molecule. And the TCR of a killer T cell is able to interact with MHC1 with the help of CD8. Review the other video for more on activation of T cells. Now we're going to jump to B cells and talk about them and what they do. So B cells make antibodies. Antibodies are proteins important to the immune response. B cells are made and mature in the bone marrow, and they're going to be found in that same type of lymphoid tissue as T cells, such as spleen, lymph nodes, MALT, and GALT. Some of the lymphocytes are circulating in the blood, and they have a B cell receptor on the surface. But the B cell receptor is just anchored antibody, as shown right here. A B cell can also rearrange their cassettes to make different combinations so they have different types of B cells. Our body can make somewhere between 10 to the 9th or 10 to the 13th different types of antibodies, an outstanding number. The variation is largely in that antigen binding area so that we have the ability to respond to many different epitopes. We can respond to pretty much anything that nature is going to show us. So here we have a B cell that has committed to making itself a specific type of antibody, and it can bind a specific type of antigen. This is a type of lock and key interaction similar to an enzyme and a substrate reaction. Whatever fits in that binding site is what is going to activate that B cell to make and secrete that type of antibody. That response is hopefully going to allow the body to clear that type of antigen found in the system. This drawing just shows how antibody is set up. Looks like a Y, right? It has a little tail and it has these little arms sticking out. To construct the functional proteins, there are two heavy chains, and which are these longer polypeptides right in the center, and then we have the two shorter light chains. Thus, a quaternary structure composed of 4 total polypeptides, 2 of a larger type called the heavy chain, and 2 of a shorter type called the light chain. They're held together by these disulfide bonds, covalent bonds between cysteines, right? The sulfur side groups on the cysteines make covalent bonds. Over on the image on the right, you can see a ribbon model that shows a more realistic view of how the antibody is shaped, how these 4 subunits of protein fit together. Antibodies do a lot of different good things for the system. Of course, I mentioned that they're complementary to the epitope. They bind like a lock and key, but they have a lot of different activities related to this binding of epitopes. In the last chapter, I mentioned that they can activate complement, and by doing that, they play a role in inflammation. They also can neutralize things like viruses or toxins. You can see in this top panel that they basically coat the surface of something, a toxin, virus, or bacteria, so that it can't bind to the receptor that it was intended to bind. By doing this, it is neutralizing the target. Opsonization, if you recall from the earlier explanation, is binding or coating in order to enhance phagocytosis. If you see down here, we have that antibody-coated bacteria. Now notice we have a receptor that fits the tail end of an antibody on the surface of this phagocyte. The tail end of an antibody is called the Fc. And there are receptors for the tail end of antibodies on macrophages and similar cells. Hence, if an antibody is coated on the surface of something, then a macrophage with the receptor for the tail end can latch on and really grab a hold of that. So now the neutralized invader cannot really escape, and it really improves the ability of the cell to phagocytose and then eliminate that antigen from the system. That explains the how and why of opsonization. Agglutination is this picture here. We can make a network by one arm of an antibody binding to one pathogen and the other arm binding another. When you get several antibodies doing this type of thing, you can group antigen together in a small network type of structure. That is agglutination, and that holds these invaders in place. So now a macrophage can come along and phagocytose a whole bunch of them at once. That is very helpful to clear out an infection. Then finally, we have this last idea, which is one of the few times we'll be talking about natural killer lymphocytes, but they too have Fc receptors. So If something is coated with antibodies and a natural killer cell happens to come by, it will latch onto that and then it will immediately dump some toxic contents out and kill the invader. We will talk later about the perforin and granzymes that causes a cell to undergo cell death, which is one possibility of this activity of NK cells. Killing of antibody-coated cells by NK cells is known as antibody-dependent cellular cytotoxicity, or ADCC. ADCC is mediated by the NK cells. My last slide for this section is going to cover the classes of antibodies. There are 5 of them. IgM is this large one that looks like a snowflake. IgM is the first antibody produced, and thus it's the first on the scene. When you start to respond, when B cells are stimulated to make antibodies and secrete them, then the first thing that they do is make IgM. IgM is the first response when you're learning to respond to an infection. You can see that it's basically 5 antibody units linked together, so there's 10 antigen binding sites. It's good at doing a lot of the things like complement activation. It can neutralize antigens, agglutinate antigens well. After IgM production, your B cells will switch. They will make something else. Oftentimes it's IgG, which ends up being the most common antibody type in the blood. IgG is good at doing all of the 5 things that we talked about: complement activation, neutralization, agglutination, opsonization, antibody-dependent cell cytotoxicity. You will find this IgG coming along secondarily as you respond to a pathogen. Also important is that after an infection, the memory cells will hang around and they will have the capacity to make IgG. So if you're measuring the different amounts of antibody in the blood, you'll find a lot of it is IgG. About 80% of the antibody concentration is IgG. Now, IgA is the type that's secreted onto the surfaces of mucosa. Here it is as a monomer, but here it is held together by this peptide as a dimer. So with this structure, you have 4 antigen binding sites. This form is secreted onto your mucous membranes, and it can also be found in membrane secretions. So you'll find it on the surfaces of your gastrointestinal tract, in tears, saliva, mucus, and importantly, breast milk. IgA is very important for protecting nursing infants. IgE is the one responsible for allergic reactions. This antibody type is not desirable so much in our industrialized society. Its true role is helping in anti-parasitic responses. But here in the industrialized world, we do not have as many parasites, and what we find is that you just have a lot of allergic reaction where IgE plays a role. That's not helpful. More on this in the future. Now, IgD is more mysterious. For the most part, people believe that it's really the form that's part of the B cell receptor, that's anchored form on the B cell surface. This would be the B cell receptor, BCR form. The other forms are secreted forms that the B cell is releasing in response to antigens. That's where we'll stop this mini lecture. We'll pick up on our next video with cytokines.